So welcome everyone, also from my side. Thank you, Tatiana, for inviting me and Ivana. And welcome everyone to this boundary-breaking science lecture today in data science. My name is Karoline, and together with Ivana, we want to introduce you today in how we're combining the power of spatial and single-cell transcriptomics to illuminate health and disease. So let me tell you a little bit about myself and how I actually ended up here where I am now. I've always been fascinated by how things are working, how cells are working, how life is actually functioning. So I studied biochemistry in Greifswald and Berlin. And after that, I pursued a PhD in systems biology, both working in the lab and on the computer. And I had a project that was focusing on method development for spatial transcriptomics, but also single-cell RNA-seq and RNA metabolic labeling. And after that, in the midst of the pandemic, I started a computational postdoc at Helmholtz Munich, excuse me, where I was working on the integration of different multiomics layers and single-cell and spatial transcriptomic data. And last year, I joined Bayer Pharma as a computational biologist. And our mission here is really to bring spatial transcriptomics into drug development and to advance therapeutics and to understand how they are working. So while Ivana's and my research projects are quite different, they have something very fundamentally in common. We're trying to close knowledge gaps here that are well understudied, and Ivana's working on the female reproductive tract, while I'm focusing on different disease areas, and one of this as cardiovascular diseases. So the female reproductive organs age approximately twice as fast as other organs due to periodic remodeling. And even the occurrence of some cancer types in women is correlated with the summed up number of menstrual cycles in life. However, there's a huge gap in reference databases, and this impedes research towards the understanding of these processes and for, of course, predicting toxicities and adverse side effects. On the other hand, heart diseases have really been a leading cause of annual death for a very long time. However, the investment in research and new drugs is comparably low, as you can see in this plot here, comparing cardiovascular diseases to, for example, oncology. And in the past, bulk technologies often, they lack the cellular resolution for mechanistic understanding of disease mechanisms. And this is really the power of single-cell and spatial technologies, which were key in this case to enable the understanding of how the reproductive cycling influences aging of the female reproductive tract, but also on the Bayer side, how diseases are working and therapeutics are working. So what is single-cell and spatial transcriptomics? How are we doing this? And basically, we start with a piece of tissue on the left-hand side. Oh, sorry. And this piece of tissue can either be dissociated into single cells, and these we can sequence individually and then annotate their cell types based on the transcriptome that we see in sequencing. On the other hand, we can use these spatial transcriptomic technologies, and this is basically taking a very thin section of a tissue, putting this on top of a glass slide, and then we sequence the transcriptome in the spatial context on this glass slide. And there are basically two different concepts in the field of spatial transcriptomics, which I quickly want to introduce here. And in one group of technologies, I would say you put a piece of tissue on a glass slide or on beads that have spatial barcodes. So when these barcodes bind the RNA, you can later know where they come from, and then build these pixelated maps, so to say, of gene expression in a tissue. And we can do this for really thousands of genes, which is really, really helpful to identify new targets. On the other hand, we have methodologies that are based on in situ hybridization. And this means that there are a couple of 100 genes that are specifically probed with small oligos. And these are read out in the fixed tissue by fluorescence at the end. And the result of this experiment looks like this. So really one dot here is one RNA molecule in the tissue. And these polygons here that you see in pink and in green are the cells that have been detected in the tissue. So you really have a very high spatial resolution, but of course a lower complexity. And with these spatial transcriptomic methods, we can see where genes are expressed, which cell types are interacting, and how these cell types react in a tissue to a certain treatment. And then in the next step, I want to show you how we enable innovation with these technologies in drug development at Bayer. So the first project that I want to show you is about cardiac disease and how we discover new drug targets and identify therapy mechanisms. So we all can improve our cardiovascular health with these life essential aids. However, we have patients with congenital heart diseases that have extremely limited treatment options right now. And by combining spatial transcriptomics with other data modalities, we increase our understanding of disease causes and the therapy mechanisms. So let me show you one example. For example, we could start with a sample like this, but this is a piece of tissue of the heart. And in this little zoom in box, you can appreciate a clustering or enrichment of these small, tiny violet dots, which could potentially be immune cells that are invading a fibrotic area in the heart. With doing spatial transcriptomics, we have these spots that cover the transcriptomes in space. We can plot, for example, marker genes for immune cells, and then really see that in this fibrotic area that we see here in the histology picture, there's a higher expression of these immune marker genes. However, in the next step, we can really take a single cell RNA data set from the same samples and use computational tools to map the single cell RNA data sets back into space. And then we can really appreciate a mapping of immune cells and subcell types here in this fibrotic area. We can see adipocytes in this particular area, which we can identify also in histology. So this is a very nice control that this is really working. And we see the largest piece of the tissue is made from cardiomyocytes. Now this is coming from one experiment, but of course we want to do this for many, many samples and different treatment conditions, right? So we need to combine different data sets from many samples and these different treatment conditions in one of these integrated data sets that you can see here. And this is a simplified showcasing of this combined data set, where one of these dots here is one spatial location in one of these samples. And we can color these dots, for example, by their treatment condition. So we have here a vehicle treatment and a real treatment, and we can really see that the content of cardiomyocytes, the percentage of cardiomyocytes on one of these capture areas is has a tendency of being higher here in this one part of the plot where we have treated samples. But of course, we can quantify this and see that under treatment, we have a higher percentage of cardiomyocytes per capture area and a lower percentage of fibroblasts, which for us, of course, is a very, very nice result. A second project that I want to show you today is coming from a clinical trial in immuno-oncology. So first, I want to tell you a bit what immuno-oncology actually is. And here we are aiming at reactivating the body's immune response to cancer cells. And this can be achieved by either blocking or removing immunosuppressive cells in the body. And again, here we start with one of these histology pictures that is actually coming from a patient from a biopsy. We do spatial transcriptomics and we can identify a multitude of cell type in these samples. We can even see subcell types, different tumor cell types, and we can appreciate how T cells, for example, which are really important for the immune response of the body, are sitting here inside of the tumor area. Now, additionally, spatial transcriptomics enables us to quantify the direct effect of our drug treatment on the target expression, which goes down in this case, and the immune response, which goes up, which is again consolidating to see. Now, additionally to these very basic analyses of the target is going down, immune response is going up, we can have a look at how immune cells are interacting in space. So when we connect all these cells that we have in space with these edges here, we get a network, and we can do the graph analyses. We can analyze these network structures and how these are changing upon treatment. And this is the so-called cellular niches that you'll probably hear a lot in spatial transcriptomics. Another important thing, of course, is how tumor microenvironment changes. So this is not only about immune cells, but also about stromal cells. One last example that I want to show you is a very, very striking example for an intricate spatial organization in the kidney. And in this case, the spatial organization is really key to the biological function of the organ. So it's really, really important to understand the complex organization on a cell type level, but also spatial. And there's a saying that we are the average of the 5 people that we spend most time with. And it's a bit similar with cells. So a T cell that is sitting inside of the tumor is having a different influence than a T cell that is sitting in a lymph node. And this is also particularly true in the kidney, where we really have a very different organization in this outer area here, which is doing the filtering of the primary urine, and then the collection that it takes place here in this middle. Yeah, and especially for the kidney, we need the cell type information to understand how a disease begins and how it progresses. And this really enables us to find new therapeutic targets. So let me briefly summarize what I talked about today. I showed you 3 examples of projects, how we advance drug development and pharma research at Bayer Pharma with spatial technologies. A cell's neighborhood really matters. And with spatial transcriptomics, we can identify cell types and their interactions in space, and spatially differentially expressed genes, which is important. And this method is dissociation-free, which I didn't mention before, but is really important for certain cell types that you otherwise lose in single-cell RNA-seq. And with this, we get deeper insights for precision medicine. So we gain a better understanding of the disease biology, the treatment response, and potential biomarkers, which are in the end important to stratify patients. And with this, I want to hand over to Ivana. Let me just stop sharing.
Okay, thank you, Karoline. I'm just going to share my presentation. Okay. So hello, everybody, one more time. I'm going to start my part of the talk with my scientific journey. So similarly to Karoline, I studied molecular biology, and after that I did my PhD at the University of Tübingen and Max Planck for developmental biology, where I studied how the microenvironment in liver cancerogenesis is regulated. So how the microenvironment influences the initiation and progression of liver tumors. Then I moved to Heidelberg, where I did my postdoc at DKFZ, and there I currently work as a computational biologist, and I study how the female reproductive system is shaped during aging, how aging influences or predisposes the system to different pathological changes, and how different perturbations of the system, such as hormonal changes, can then influence the aging trajectory. So how they influence normal aging, if they speed it up or they slow it down. And this research is something I'm going to tell you a bit more about today. So as Karoline already mentioned, there are two unique characteristics of the female reproductive system. First is that the female reproductive system ages faster compared to somatic tissues. When I say it ages faster, I mean it loses its function faster. So women and other females of the mammalian species lose their ability to reproduce as they enter menopause or equivalent of the menopause. A second unique characteristic is that female reproductive system undergoes cyclical extensive remodeling. So this happens as a preparation for the pregnancy, and in each cycle, there's a lot of proliferation happening as a support for anticipating pregnancy. And if that does not occur, the whole system resets itself and the new cycle starts, where again extensive remodeling starts. In women, there is an epidemiological observation that the number of the reproductive or menstrual cycle is linked to the risk of uterine cancer. So the higher number of cycles, the higher the risk. Since cancer is disease of aged tissue, we asked in our research if there's a link between then cycling and aging. So what type of relationship exists between these two phenomena? To answer this question, we use single-cell RNA-seq and spatial transcriptomics technologies. So Karoline already explained to you in the beginning how these two technologies work. So in our case, we use these technologies to isolate the organs of the female reproductive system, so ovary, oviduct, uterus, cervix, and vagina. And we then prepared single cells and sequenced them. We did this from young mice and from aging mice. So I would just like to briefly comment why we use mouse as a model organism in this case. So what we are doing in our research is that we are studying normal aging. That means that we would have to collect tissues from healthy women. Since there is no medical reason why we would need to take biopsies from healthy women, it would be unethical to do that. Second alternative, which we could do, for example, would be post-mortem. However, the cohort of patients which we would have to have then would be enormous, because we have to control for all the factors that can influence reproductive cycles, such as hormonal contraception, pregnancies, breastfeeding, smoking, BMI, and all that during the lifetime of a woman, because we want to know what's the relationship between the number of cycles during the lifetime and aging. So since then cohort would have to be enormous, and the research at single cell and RNA-seq and spatial transcriptomics is quite expensive, with mice with much smaller number of replicates, since we can control the conditions much better, we can get much cleaner result. And that's the reason why we use mice. So we isolated then the reproductive organs of the reproductive system from young mice during all phases of their reproductive cycle. So reproductive cycle in mice is called estrous cycle, and during pregnancy. We did this similar thing during aging. So we isolated these organs at 9 months, 12 months, 15 months, and 18 months. So just to briefly put the aging of the female reproductive system in mice into perspective. So mice at around 12 months are in perimenopause, or they're entering perimenopause. At 15 months, they are already starting to enter menopause, and at 18 months, they are in menopause and they're not cycling anymore. Secondly, we collected ovaries and uteruses from young and old mice and performed spatial transcriptomics on these organs. Okay, so in our research, we focused on two processes, which are known to increase their activity during aging and which are linked with different pathologies such as cancer. So these two processes are inflammatory response and extracellular matrix reorganization. In our research as well, we focus on fibroblasts as the cell type which might be responsible for then the changes in these two processes during aging, since fibroblasts are the cells which certain research earlier showed might have inflammatory memory, and they're as well the main source of the extracellular matrix. So what we first did is using our single-cell RNA-seq, we performed differential gene expression and try to profile which genes are differentially regulated during the reproductive cycle in mice in different organs. After that, we did overrepresentation analysis to know which pathways these genes, which are differentially regulated during cycle, regulate. So these are the results for different organs. So on the columns are the organs, on the rows are the pathways. You can see, so here we wanted to have a sanity check and see that actually inflammatory responses such as TNF-alpha and different extracellular matrix pathways are differentially regulated during the cycle. And that actually is the case. So we see that these processes are popping up as being differentially regulated during cycle in different organs. Second, what we wanted to know is how the activity of these pathways is modulated during the cycle. So this you can see here in the second part of the slide. So the first plot is inflammatory response, second plot is the extracellular matrix response. On the x-axis are the phases of the mouse cycle. So we start with proestrus and end again with proestrus. And on the y-axis is the activity score of these two pathways. Each line represents one organ. And as you can see, in different organs, there's a different pattern. So for example, vagina, oviduct, and cervix follow this inverse U shape during the cycle. The activity of the inflammatory response follows an inverse U shape, while uterus and ovary follow U shape, and they're much less regular. They're much less, changed compared to the other three organs. When it comes to extracellular matrix organization, uterus, which is in pink, shows the most extensive remodeling of the extracellular matrix. So this we anticipated, since uterus is an organ which has to support pregnancy, and because of that, it remodels most during the cycle. Second thing that you can see from here is that although these organs are all under hormonal,
it's where we stopped, or maybe
yes, so this was transition to the next slide.
Right, okay.
I'll skip one slide because of the time constraint now. So this is what we used our single cell RNA-seq data. But as Karoline already explained, in the single RNA-seq data, we lose this spatial component because the cells are dissociated from the tissue. So we don't know then what's the environment around each individual cell. So to actually look at how cells which have high inflammatory activity, so fibroblasts which have high inflammatory activity, are located in the tissue, if they are uniformly dispersed across tissue or they form clusters, so some inflammatory niches, we used our spatial transcriptomics data. So we again measured for different fibroblasts in the spatial transcriptomics data, their inflammation score, and then we binarized this score. So we said the cells which have the highest 25%, the cells which have the highest 25% of the inflammatory score are considered high stromal cells, so high inflammation stromal cells, and the rest are considered low. And then we built the spatial graphs to try to determine more high throughput manner if the cells are forming isolated spots, like for example, you can see here, or they're actually forming something like clusters here. So how we build the spatial graphs is that if we have highly inflamed spots, fibroblast spots, we just connect them with an edge. So if there are two neighbors. So for example, here, if you have two spots which are direct neighbors, they will be represented in a graph as two spots connected with an edge. If it's an isolated spot, then it's represented just as an isolated spot, etc. And then you can sort of quantify this by determining how well connected your dots in your spatial graph are. As you can see, quite often from already by an eye from this ovarian section, quite often these spots come in clusters. So that's how they're represented in both ovary and in uterus. You can see that's by the level of the score, because if the score is 0, then there's no connection between the graph, and if the score is up to 1, then the spots are completely connected. The second question we asked here, okay, so they tend to form clusters in the tissue, but do these clusters tend to increase as you age and as your inflammation level increases? In the ovary, we don't see much of an inflammation increase as you age, and we don't see as well an increase in the cluster. While in the uterus, where we see general inflammation of the fibroblast increasing with age, we as well see an increase in the age of the cluster, if the size of the cluster, meaning that once you have these inflammatory spots there, as you age, as the inflammation increases, they tend to just spread. So some spots will be form anew, but a lot of them are spreading, meaning that it's probably then the other fibroblasts, which are in the inflammatory niche, are the ones who are actually supporting them and propagating the inflammation during aging. So just to go back to our original question, then what is the link between cycling and aging in these two processes? We determined the modulation of the inflammatory activity during the reproductive cycle in mice, and that's shown here on the x-axis, and then age-related inflammation for fibroblasts and for the immune cells. You can see that in fibroblasts, there's a nice relationship between the modulation of the inflammation during the cycling and age-related inflammation, meaning that tissues that do tend to modulate a lot the inflammatory activity tend to end up with a lot of inflammation in the old age in fibroblasts. With immune cells, we don't see this relationship, meaning that the fibroblasts are the cell type that actually is retaining this inflammatory memory. We see a very similar relationship for the extracellular matrix. So if there's a lot of remodeling during the reproductive cycle, you end up with a lot of fibrosis or a lot of extracellular matrix deposition in the old age. So how do we interpret this? So let's say, for example, let's take uterus. So uterus will remodel its tissue a lot during the reproductive cycle. So this has to happen as an expectation or so, as a support to pregnancy. So at the beginning of the cycle, uterus is very, very thin. As the cycle progresses, uterine tissue will proliferate a lot. You have a lot of extracellular matrix, so this mesh, which is between the cells, is being produced. This is necessary because in the second part of the cycle, if the pregnancy does occur, it needs to support the growth of the implanted zygote. If the pregnancy, however, does not occur, this tissue has to degrade, and then in the new cycle, again, the whole thing starts. So you'll have at the end of the cycle very thick tissue, which is degrading, and women usually then have the menstruation. At the beginning of the cycle, you have again very thin tissue. Now you can imagine that this happens during lifetime, and in each cycle, there's a bit more of this extracellular matrix, which is not completely degraded. So just a small percentage. And as you cycle then with the number of cycles, that actually increases during a lifetime. So this was our hypothesis, and from this relationship, it was hypothesis based on the sort of correlative relationship. We wanted to show that this is really that there is really a causality between the number of cycles and fibrosis in the old age. So the problem with this is that cycling and aging are exactly collinear, meaning that they happen in the same time. So to be able to distinguish what's an effect of cycling and what's the effect of aging, you need to stop one. We cannot stop aging, unfortunately, in this moment. So we stopped cycling. So we have two groups of mice. First is the control group, where mice are cycling and aging. And the second is VCD group, where we administered to mice a chemical called VCD. This chemical pushes mice in menopause, and they only age, so they don't cycle anymore. And we compared then the fibrosis level in the oviduct and the uterus. And you can see that indeed in the mice which only age, there's much less fibrosis in both organs compared to the mice that cycle and age. So that means that the cycling is directly contributing to the fibrosis by having this incomplete resolved ECM process during each cycle. So if you cycle and age, you have a lot of fibrosis. If you just age without cycling, you will have much less fibrosis. So just as a take-home messages, single-cell RNA-seq and spatial transcriptomics can be used to provide insights how specific cells, individual cells function, and to profile interactions between these cells in the microenvironment. We use these technologies to show that this remodeling, which happens during each reproductive cycle in the reproductive system, has actually unexpected cost and creates unfavorable microenvironment. And I would just like to thank all the people who participated in the project and you for your attention. Thank you.